Okay, so the final method of molecular subtyping that we can use in cancer detection is multiplex ligation dependent probe amplification. And this is another technology that absolutely requires a sound understanding of polymerase chain reaction before you can understand how it works. So if you've not done the PCR tutorial yet, go back and do it now. So MLPAs are used for mapping large chromosomal deletions and also chromosomal imbalances, and that's ideal for things like the HER2 gene locus in breast cancer, where you get gain of chromosomal material. We can also use it for multiple oncogene gains and tumor suppressor gene losses in cancer in a single reaction. And basically what we can do with this assay is detect up to 40 different chromosomal locations and quantify whether there's chromosomal loss or gain at that location. And what we do is we perform a PCR amplification and we detect it using capillary electrophoresis instead of a DNA gel. And this is a sort of technology used in DNA sequencers. I'll not burden you with that particular technology just yet. So this is the basics of how MLPA works. MLPA works by having multiple probes, and those probes bind to target DNA. So target A could be the HER2 gene, target B could be a different gene. And each probes come in two halves. So that's one half, that's the other half. And if they successfully bind to the target, they become ligated together with DNA ligase. So we allow these probes to bind. DNA ligase fills in the gap and creates a piece of DNA from here all the way along to here. And we do this at up to 40 different locations throughout the genome, so we end up with 40 completely new novel pieces of DNA after this ligation has been performed. Now each of those ligated pieces of DNA are different lengths based upon this, what we call stuffer sequence here, which is a different length in each individual probe set. So this could be 20 base pairs in length, the target region might be 20 bases in length, this might be 20 bases in length, the stuffer sequence could be 50 in length, and then you've got another PCR primer binding site here. So the whole length here, maybe as I've said there, is 130. This target B could be 150, target C could be 170 base pairs in length. So probe set A recognises target A, and this bit in purple is perfectly homologous to the target DNA. Target B, this bit here, matches the target DNA. Where they're identical is this bit here is identical to this bit here, because this is a PCR primer binding site. This black bit here is identical to this bit here, because this is also a PCR binding site, and this green bit differs on every single probe set to make the final product a different length. So these slides show how a reaction occurs. You initially denature your DNA and hybridize your probe sets. And remember, each probe set comes in two halves. So if the forward bit and the reverse bit stick together, or bind here, DNA ligase comes along, links the two together to create a piece of DNA from here all the way to here. Same with target B. So that's created two completely new pieces of DNA. So this is what these two new pieces of DNA will look like, and they both have PCR primer binding sites on the end. So both pieces of DNA, even though the bit in the middle is different between here and here, completely different sequence, they are both amplified by the same primer pair. The forward primer binds here, the reverse primer binds here. So and I'm only showing two probe sets here. You've got 40 probe sets in a typical MLPA. I'm just showing two of them, but all 40 are amplified with the same primer pair. And the PCR products themselves are detected by capillary electrophoresis, and that they can differentiate between different size products, and we get a trace that looks very much like a HPLC or liquid chromatography trace. It looks like that. It's just a trace, and we are detecting DNA as it comes off the column, and each of these are different amplified PCR products. The forward probe can be fluorescently labelled, and that makes detection much easier as the DNA comes off the column and passes, let's say, a UV or a fluorescence detector. So to summarise the whole procedure, you've got target A, you've got target B, you've got target all the way up to target 40. Two halves of a probe pair bind adjacent to each other. The gap is filled in by a phosphodiester bond by DNA ligase. That can only happen if this is bound on a piece of DNA to this. If these are just floating around in solution, DNA ligase can't link them together, and that's the crucial step. We then generate, we then denature that DNA, and we've got bits of DNA like this, which represent the two probes bound together, and then these are PCR amplified by using a single set of PCR primers, and that gives us our 40 different pieces of DNA, all of different sizes, and the different sizes due to the stuffer sequence being different lengths, and then we separate them by capillary electrophoresis and quantify them. So summary of the protocol is here you heat denature your DNA, you hybridize your probe. This is Salsa probe mix is the commercial name for these. So you hybridize the probes to your extracted DNA, incubate for a minute at 95, 16 hours for the DNA to hybridize to the DNA, that's conveniently overnight. Add your DNA ligase, and that will link the two probes together if they are bound to the same piece of DNA. Then heat inactivate the ligase, and then you perform your polymerase chain reaction on these ligated products. Perform our PCR amplification. After the completion of PCR, we run that down capillary electrophoresis to determine the size of each product and compare to a reference sample for relative abundance, and that tells you how much target DNA was present that those probes bound to. So this is shown here, and we can use this for screening regions of DNA. In this case, we're dealing with the APC gene. This is a gene that is commonly deleted in colon cancer, and we sometimes want to find out whether cancers are deleted for specific regions of this gene. Now, quite often it's only certain exons that get deleted. So this is an MLPA control DNA versus an MLPA cancer, and all of the peaks look very similar between the control and the cancer. Same peak height. You know, this one's higher than this one, but that's okay because in the cancer it's exactly the same. So if you follow the pattern of the peak heights, you see that this one and this one are the same, but this one, and this one are about half the height of what they should be. And that's because in this particular cancer, one copy of the APC gene has been deleted for exon 11 to 13. These probe sets are in exon 11, 12 and 13. It looks like the rest of the gene is intact. So you can use this for quickly screening exon deletions and what we call intragenic deletions within large genes. So you can buy entire probe sets that cover every single exon of your favoured particular gene. We can also do this for analysing just chromosome abundance and chromosome number. And if you think of how many chromosomes we've got 22 chromosomes plus X and Y. That's perfectly reasonable to have an MLPA set on every single chromosomal arm, and then you can get relative quantification of chromosomes in a DNA sample. So in this particular case, which was a prenatal test, here you've got a male reference sample, a female reference sample, and you can see that the X is higher here than here because this is XY, this is XX, and this particular sample is a triple X. So this is a fetus which has got 3 X chromosomes, and we can spot that because this particular peak height is higher than here. So you can do that for all chromosomes. If it was Down syndrome, you'd see the chromosome 21 peak being a little bit higher. So where this test is really useful is screening lots of oncogenes and lots of tumour suppressor genes in cancer in one simple reaction. So this is a breast cancer sample, and here's a reference sample, and here is a breast cancer sample that's got BRCA1 deletion, and we can see here this just happens to be the peak that refers to the BRCA1, and so does that, which is showing that the reference peak is here. This peak is much lower, and this one is much lower than expected there. So that's telling us that the BRCA1 gene is missing in this particular sample. A lot of the other genes in there are present at normal rates. So there are some losses and gains. I'll not go into them, and that was just one example. In this example, we've got amplification of the HER2 oncogene. HER2 is also called ERB2. So all of these here are, there is no, I'm not showing a reference sample particularly here, although it can be compared to this one, but the scale is completely different because this is a gene amplification which is much, much higher than all of the other peaks. So what we can see is this highest peak here is ERB2, so we know it's HER2 amplified. This is also HER2 here, and this is also cyclin D, NeuroD. It's just a different name for it. This is cyclin D. So this particular cancer has got cyclin D gene amplification and HER2 amplification and something else here that I'm not entirely sure what it is. So the MLPA assay is a very quick, well, it's not particularly quick, it's a nice technique for testing lots and lots of chromosomal locations. That can be lots of different genes, it could be lots of different chromosomal arms, it could be even exons within a single gene, and test them all in one simple assay and get relative quantification. Is there more DNA or less DNA than a reference control sample? So that's where we use it for screening that type of situation. So this is a summary of everything that we've covered. We've covered FISH. This is useful for detecting a single amplified or deleted gene region or an entire chromosome. We've looked at CISH, which is a chromogenic version of that. We've looked at minimal residual disease testing. We've seen how qPCR is the most sensitive. Immunophenotyping, I'm not giving you much detail on that, but that's somewhere in between. FISH and karyotyping are only any good for diagnosis, they're no good for minimal residual disease testing. And then the final bit is the MLPA bit. MLPA, you can perform a screen for 30 or 40 different regions of the genome. You ligate two probes together using two half probes. They are ligated if they bind to target DNA by DNA ligase, and then you detect when those ligated probes have been ligated together. If they don't get ligated together, it's because they've not bound to the target DNA, and therefore PCR can't amplify them. So that's a whole range of different molecular techniques, two of which are utterly dependent upon understanding PCR, which is MLPA and quantitative PCR. FISH is complementary to these techniques for various applications, whether it's diagnosis or minimal residual disease testing. But all of these techniques are used routinely on cancer samples. Okay, that's the end of this session.